Okay, good evening, everybody. Welcome to week 3. We will be waiting for some time for around 2-3 minutes for everybody to join in, and then we can begin. Uh, I hope I'm audible to all of you.
Good evening, sir. Yes, you are audible.
Okay. Okay, good evening, everybody. I am Sanket Mukherjee, a Prime Minister's Research Fellow from the School of Biological Sciences, IIT Delhi. Welcome to the third week's live interactive session. This is week 3 of the 12-week course of Genome Editing and Engineering, which is taken by Professor Utpal Bora of IIT Guwahati. Um, I think most of you have joined in, and we will be beginning in just one more minute. Um, yeah, so let's begin. So in week 3, what has been essentially discussed is homologous recombination and non-homologous recombination. Is the slide changing? Okay, now I think it is. So homologous recombination mostly and non-homologous recombination as well, that is non-homologous end joining, which are essentially, we have discussed it in the previous week as well. Homologous recombination and non-homologous end joining are two fates when there is any double-stranded break in DNA, right? These are the two pathways where it can go. We will also discuss about site-specific recombination, which is very similar to homologous recombination. It is essentially recombination of genetic elements of genes in a genome. Now, ideally, these all processes, why are we learning these? Because they are present naturally in biological systems. And essentially, this course of genome editing and engineering, in this course, we will learn how these natural processes are exploited to our own advantage. So if we want to introduce a gene of interest, if we want to delete a gene of interest, we might uh, refer to some of the enzymes which are naturally used in the DNA repair process, or in case of recombination, there are various recombinases. We'll discuss about it today. So just to recap what we have already covered about DNA damages, right? Just let me just give me a moment. Yeah. So DNA damage can be of two types. One is endogenous, another one is exogenous damage. So we all know what these damages are. Uh, so endogenous damage is in when within the cell there are some processes which happen within the cell. There are factors within the cell. If they cause any DNA damage, that is endogenous damage. So there majorly it's because of the reactive oxygen species which is produced due to metabolism, catabolism, anabolism both, like processes, cellular processes like respiration that causes generation of reactive oxygen species. And there are several metabolic byproducts which are um, which are categorized as reactive oxygen species. These reactive oxygen species are essentially free radicals, which can, interact with DNA and can damage them, right? Other reason for endogenous, DNA damage could be replication error. So it's very simple. It's essentially the DNA polymerases are present inside a cell, which um, which replicates the DNA, right? That we have already studied. Now, these DNA replication, these DNA polymerases, they have varying fidelity, right? All DNA polymerases are not as accurate. So some of them may cause some errors, right? This is what we already know. Then there could be DNA base mismatch. So mismatches could occur due to a problem in the DNTP homeostasis. There could be several other factors. There could be some A-basic sites. There could be analogs of of the nucleotides. These nucleotide analogs, they can also cause incorporation of DNA mismatches, right? Then there is like the topoisomerase DNA complex. So topoisomerases are the enzyme which are responsible for DNA supercoiling, basically the DNA structure and organization in a cell. Topoisomerases are responsible. Example, helicases. So they unwind DNA. So DNA topoisomerase DNA complexes could introduce some errors inside DNA, right? In a cell. Now, some exogenous damage, the most common one is the UV damage, which we already know. UV rays, X-rays can essentially damage DNA. UV has been implicated in causing multiple forms of cancer as well. There could be some hydrolysis reactions which might happen inside a cell, which can cause even DNA to be damaged, right? Another exogenous factors which could be responsible would be some plant toxins, other kind of toxins which are not naturally present inside the cell, but these are exogenous factors which could damage DNA, right? Now, there are five major repair pathways for these DNA damages. We have already discussed about these. So just to recapitulate, one is base excision repair. When there are lesions in on a single base level, base excision repair repairs them. If there are bulky lesions, then there is nucleotide excision repair. If there are mismatches which are happening, there is mismatch repair. Okay. Then there is homologous recombination and non-homologous end joining, which are essentially the repair pathways which are undertaken by the cell when there is a double-stranded break. Now, a problem in any of these repair pathways could also lead to DNA repair, DNA damages, right? And essentially, these are implicated in various diseases. You have already studied about them. So that's it about DNA damages. Now, so this slide talks about the DNA repair mechanism. Can you see the slide of DNA repair? Something is wrong with the slides today. Yeah, at least I can't see them as, yeah. So base excision repair, if you remember, it involves DNA glycolases. These DNA glycolases cleave DNA at the N-glycosidic linkage, right? And there is then production of an A basic site, and then this A basic site is further, repaired, and then there is ligation of the damage, right? Then there is nucleotide excision repair, where if there is a bulky lesion, it is removed and then it is repaired. Gap filling is done. In case of mismatch repair, if there is a mismatch, this mismatch is removed, and then there is synthesis and ligation, which is common in almost all the repair processes. Non-homologous end joining, we had just briefly discussed about it. It's there we have the uh, Ku proteins or Ku proteins, right? Which are important, which to recognize the DNA damage, double-stranded breaks, and then there are multiple accessory proteins which come in play, and then they essentially ligate the two ends of the double-stranded break, right? That is what happens during non-homologous end joining. Multiple insertion deletion, usually deletion occurs because it's not a process with a high fidelity. If we talk about homologous recombination, it's much more common, it's much more conserved in eukaryotes and prokaryotes. And here this is basically one of the major pathways how double-stranded breaks are repaired, right? So here what happens, there is strand resection, after which the affected chromatid, it finds homology with another chromatid which has the same sequence. And based on the other sequence, there is formation of holiday junction, this strand invasion and holiday junction, and it causes, you know, polymerization and production um repair of the damage, after which there is resolution of this complex with which we get the normal DNA. So these are the five major processes. Now, double-stranded breaks, if we talk about the double-stranded breaks in general, right, they are caused by endogenous stresses, like there could be reactive oxygen species which could lead to double-stranded breaks, right? That is one of the ways. There could be replication errors as well. There could be DNA adducts which are formed which would lead to DNA double-stranded breaks. There are multiple reasons for double-stranded break. These are very toxic, right? And they need to be repaired as soon as possible. Also, these damaged double-stranded breaks are usually one of the most common forms of damages that happen in a DNA. Now, there could be exogenous factors also. For example, UVB damage could UVB radiation that could cause double-stranded breaks to form. Another thing that could be there is, as I already said, errors in DNA replication, which comes under endogenous factors only. Now, it's not always true that double-stranded breaks are damaging. It's not always like that. In some cases, it could be beneficial. So in what cases could it be beneficial? For example, during meiotic differentiation, some genomic rearrangements are they happen due to double-stranded breaks. So essentially, the formation of breaks and repairing them together can essentially bring about some variations in the genome, and which is very important for cell survival and some other processes. Now we will talk a little bit about the strategies of double-stranded break repair, right? So one of them is homologous um, recombination. I think it's there here. Homologous recombination. Um, it's a high fidelity process. So this process is homologous recombination. We have already discussed this in the previous lecture as well. So homologous recombination could be one fate of repairing a double-stranded break. The other one could be non-homologous end joining, right? Non-homologous end joining. Now, non-homologous end joining could also be um, like there could be like the canonical form of non-homologous end joining, or there could be alternative forms. So before getting into homologous recombination, what essentially happens is there is end resection. Basically, the ends of the double-stranded break, they are resected using multiple proteins, and then there are other proteins which are involved, which basically cause strand invasion and then repair. We will talk about it in details. What is important to know here is that homologous recombination is a high fidelity process. So it's usually more accurate in repairing a DNA damage. And if there is a damage in one chromatid, that is repaired using its sister chromatid, right? Which is usually not damaged. So one of the, if we know that we have a diploid genome. So if one of the chromatids are affected, the other would be probably fine, and we will this will be the homologous template, which will cause the damage repair pathway to work. If we talk about non-homologous end joining, there it is an error-prone process. It's an error-prone process. Usually when the damage is too high and there is no homologous strand present, or the machineries or the associated factors for homologous recombination, those are not present. At those times, non-homologous end joining happens. Usually this causes small deletions, right? And it's very simple. It's basically joining two broken strands. They are joined. In case of alternative form of NHEJ, there, I mean, non-homologous end joining, it causes more damage because it causes larger deletions. Now, talking about the key steps in double-stranded break repair. So either using the MRN complex or the KU70 and KU80 complex. These are complexes which recognize a double-stranded break, right? So if the MRN complex binds to a double-stranded break, let's say this is a DNA, which a double-stranded break. Now there are two fates to it. Either MRN will bind to it, or there will be KU70 or 80. So Ku complexes will bind to it. Based on this, it will decide if the MRN complex binds to it, it will go towards homologous recombination. If the Ku proteins they bind, then it will go for non-homologous end joining. So in case of homologous recombination, we have seen there is a short range resection. This is the protein complex which causes short range resection. And Uh, then there is long range resection also, which we will see very soon. And then there is RPA, which is another protein which is very important for strand invasion. We will talk about it in detail. So if we talk about homologous recombination, it is basically what is homologous recombination? So it's pairing of identical or nearly identical, right? Strands which would cause an exchange of genetic material. Okay? Homologous recombination is it's so important in a genome that it is also called the guardian of genome integrity. This is very important because it's basically conserved across prokaryotes and eukaryotes throughout evolution. This process has been conserved, right? Um Homologous recombination are involved in rescuing replication forks that have been. So in case of replication forks, you know how replication forks look like. So it's like this. At times when the polymerase stalls, there is stalling of the polymerase due to some damage. There could be like missing factors, right? There could be problems in the fork. There could be supercoiling. Multiple reasons could be there. When the replication stalls, at that time, homologous recombination happens to rescue this kind of a problem, right? If we talk about, I've already told you multiple times that in prokaryotes and in eukaryotes, this is conserved. For example, RAD51, which is a functional and structural homologue of REC-A. So these are present in prokaryotes and eukaryotes respectively. Uh, Essentially, homologous recombination has been linked to evolution of multiple genes. So in cases when genes are duplicated also, they have some variations in genes. These kind of events have happened due to homologous recombination. Okay, so there could be a change in the copy number of the gene. So if a gene is present only like once, it could be multiplied, like it would be present in more than like 2 times, 3 times, sometimes 4 times. These kind of events happen due to homologous recombination. There could be deletion also, which could happen. There could be duplication, amplification, all these things, right? Uh, if there is intrachromosomal recombination, that means in the same chromosome, if there is recombination, that leads to uh, intrachromosomal recombination, that leads to duplication basically. So the same strand could basically act as a template as it, coils down and it forms the template for the same, I mean, damage in the same strand essentially. So there could be a duplication or there could be deletions as well. So there are multiple regions in the genome which are present as duplicates, or which are present as which are usually deleted, or even they are present as duplicates. For example, telomerase regions. There are other regions. There are some satellite regions which are involved. Why knowing about homologous recombination is important? Because it has some applications also. We can fine tune this process for gene therapy, where we want to introduce our gene of interest inside the cell, right? So this is what it is all about. Now, this is essentially, this slide talks about the same thing, that homologous recombination is the guardian of genome integrity. So RAD51 is the structural and functional homologue of bacterial strand exchange protein, REC-A. Homologous recombination is a highly conserved process across prokaryotes and eukaryotes, and it's the driving force of evolution in multi-gene families. Now, what are the molecular model which is involved behind homologous recombination is strand exchange. branch migration and resolution. You can see this in the figure on the right, that whenever there is 5' to 3' resection, after that there is strand invasion, where the damaged strand invades the the homologous strand which is not damaged. So the blue one here is the undamaged strand, and the red one is the damaged strand. So the damaged strand would invade the homologous strand, right? That is how it happens. Then there is branch migration, and new DNA is synthesized based on this template DNA. And essentially, the very big, I mean, the end resection is performed so that the end could be could act as a primer for the polymerization to happen. This kind of junction, when there is uh, you know, uh, like recombination is happening, this kind of a junction is called a holiday junction, as it was discovered by Holiday scientist. And then it is resolved to form two separate DNA strands which do not have any damage. But in this process, there is some exchange of genetic material. So that's how it works. So if we talk about strand exchange, what is strand exchange? Strand exchange is when the broken DNA pairs with the homologous region of sister chromatid. Then there is strand invasion, which leads to formation of holiday junction, right? Uh, so there is a heteroduplex DNA which has DNA strands from sister chromatids. Now, holiday junction is translocated along DNA, extending the region of heteroduplex away from the initial crossover site. And then the holiday junction intermediate is resolved. So resolution is the last step which happens. And then there is a cleavage, which will basically separate. So holiday junction looks something like this. So if it's a DNA, this is how it looks like. So almost perpendicular to each other. So this complex is then cleaved later, and it will lead to generation of two separate DNA. But these are recombinate. I mean, there is recombination. There is some exchange of genetic material in the process. So REC-BCD, this is the enzyme complex in prokaryotes which are involved behind double-stranded break repair processing. So it's a bipolar helicase nuclease that splits the duplex and digests them into until a chi site is encountered. We'll talk about it, what it is. Chi site, which with this is the conserved region in the chi site, is the recombination hotspot. This is the sequence, and usually this sequence is also conserved in a chi site. Essentially, recombination is where it begins, because based on this sequence. Upon entering a chi site, the nuclease activity is attenuated. So REC-BCD, it has nuclease activity and it has helicase activity. So there the nuclease activity is attenuated, and REC-BCD, it loads REC-A, which causes further double-stranded break repair processing, right? This is all about it. You can study about the REC-BCD enzyme structure and function, everything. So if you look at it, so REC-B, this is the heterodimer, as you can see. It cuts where it continues unwinding DNA and loading REC-A. So essentially, if we look at it, in a REC-BCD complex, the REC-C recognizes the chi site. This is the chi site, as you can see. The REC-C is the enzyme which first recognizes the chi site, after which REC-C will signal REC-D to stop. When stopped, the REC-D complex signals REC-B to cut DNA. If you remember, Reg B has two activities. It has dual function. One is nuclease, which helps in cutting DNA. Another one is helicase for unwinding DNA. So Reg A, when it is loaded, this nuclease activity is attenuated, and then it functions, it processes for further, DNA repair process. We will look at it, right? Later. Am I missing something? Let me check. Okay, fine. In case of, this was about prokaryotes. So Reg BCD complex is in case of prokaryotes. Its homologue is RAD51. We have already studied. So what happens? There are sensors essentially which sense the double-stranded break. These sensors are mRNA complexes, which essentially stands for three group of proteins: MRE11, RAD50, and NBS1. Okay, it is called the mRNA complex or MRE11 complex also. So it leads to rapid activation of ATMs. ATMs are ataxia-telangiectasia proteins, which is basically a mutated kinase, right? It's a mutated kinase which will cause phosphorylation of various proteins. Also, out of which BRCA1 and P53 binding protein, that is 53PB1. These two are essential, which to decide where this double-stranded break repair will go to. So if it is 53BP1, then it goes towards the canonical non-homologous end joining pathway. And if it goes to BRCA1, then it goes for the homologous recombination repair pathway. So these two complexes, they decide with what will be the fate of the repair, right? Uh, Apart from this strand resection function, BRCA1 is also involved in loading another very important protein, that is RAD51. We will discuss about it. Okay, about it. Now, if you remember, BRCA1 is also an oncogene. Any damage in this kind, sorry, it's also important in cancers, especially breast cancers. So if there is any damage in this gene, it will impair the DNA repair pathway, out of which there would be multiple accumulations of DNA damage that leads to cancer essentially. So this is why the importance of DNA repair is very, very, very important. I mean, in terms of various diseases and also in uh, you know uh, in genome editing as such. So what happens now? This RAD51 is loaded on a single-stranded DNA, after, which happens due to BRCA1. We know about it. Now it searches for an homologous strand. Now, usually a protein called RPA, which is replication protein A, it protects the single-stranded DNA. So if a double-stranded DNA is then converted to a single-stranded DNA, there are multiple proteins of RPA1 which protect this from any damage, because single-stranded DNA is usually less stable, right? Now, BRCA1 is the protein which facilitates replacement of this RPA1 with the uh, RAD51 protein. Now, when this RAD51 protein is attached, let's say this is how the protein looks like. It's a little bit different from uh, RPA1. So when RAD51 is attached to a single-stranded DNA, that is when starts searching for homologous strand, right? We'll look at these processes in detail later. So then there is a D loop formation. And then there is DNA synthesis, which happens due to numerous polymerases. And then there is this homologous recombination intermediate. It is resolved. So resolution is the final step, which happens, out of which the repair finally, takes place. If we talk about non-homologous end joining, I already told you there are two pathways. One is canonical. So in case of canonical um, pathway, it is dependent on Ku heterodimers and DNA Ku7018 specific and DNA PK catalytic subunits. So there are several other, proteins also which you should study in detail. Several nucleases are present, which help in the non-homologous end joining. There is TDP1, 2, and Artemis. These are some other proteins which help in removing hairpins. So essentially, when a DNA is damaged, it can form a hairpin-like structure like this because of base pairing, which is not a normal base pairing. It could cause formation of hairpins. So these kind of hairpins, and then there are some adducts which are formed, which are basically addition of some chemical groups to this DNA. They all are repaired using I mean TDP1 and Artemis proteins, and then these ends are then rejoined by ligases LIG4, XRCC4. These are some protein complexes, and XLF complex. Uh, proteins are very, very important proteins. These are also very conserved in prokaryotes and eukaryotes. This is also very important for maintaining the genome integrity. Now, in eukaryotes, we have the Ku70, Ku80 heterodimer. So 70-80 is based on the molecular size. And when there is Ku protein is bound to the double-stranded to the DNA ends of a double-stranded break, it improves the nuclease, polymerase, and ligase components. So essentially, Ku proteins discover this double-stranded break, and once they discover it, the rest of the processes are carried out much smoothly. Another way of non-homologous end joining is alternative non-homologous end joining. This is a very rare process, very uncommon process. Um, so this simply means when two ends are joined, right? When two ends are joined, they are called, that is basically alternative homologous end joining. It is the slowest process among all the processes. Like if we compare like homologous recombination, if we talk about canonical non-homologous end joining, compared to these two, alternative non-homologous end joining is the slowest. And when none of the processes work, when the damage is so extensive that none of the processes work, at that time, alternative non-homologous end joining comes in picture. So this is all about it. So here I've prepared some slides on canonical non-homologous end joining. We have already talked about the sensing process, which happens due to Ku heterodimers, which further it provides a docking site for DNA PK catalytic subunit. Then the damage is processed through nucleases such as TDP1, 2, and Artemis, which removes hairpins and damaged bases and protein adducts, DNA protein adducts. Then there is ligation, which is carried out by LIG4, XRCC4, and XLF complexes. DNA synthesis happens after this. You can follow it up in the right side of this slide as well. DNA synthesis, where essentially the 3' strand, it acts as the primer, and then there is polymerase delta, which performs polymerization in this case usually. Then the homologous recombination site is sorry is this site is then resolved. Yes. In case of alternative end joining, when both the homologous recombination and canonical non-homologous end joining doesn't work, at that time, this is the route which is preferred, right? Some complexes are involved, um, which are involved in homologous recombination and non-homologous end joining that is shared by the alternative end joining process. It is the most mechanically distinct repair pathway and has been shown to be active throughout the life cycle. There is not a particular phase where it is preferred. It is essentially enhanced in G2 compared to G1 phase, and is in compromised state in case of stationary phase uh, of the cell cycle. Then we will talk a little bit about site-specific recombination. So site-specific recombination is essentially a recombination event which is not homologous recombination. So there are various recombinases. These are enzymes which essentially causes this site-specific recombination. So what could they process DNA molecules and they rearrange them by breaking and rejoining the strands at specific points. They are used for targeted, gene alterations. So for example, there are three processes which are discussed. One is excision, where your gene of interest could be removed completely, right? That could happen due to recombinases. There could be inversion, where the gene of interest is inverted. Okay. And then there is insertion also, where if you see that the gene of interest is inserted into your site of choice. So these are the three main processes which could happen with site-specific recombination. If we compare the homologous recombination versus the site-specific recombination, HR versus if we talk about site-specific recombination. So homologous recombination, we already know it. We need a double-stranded break for this process to happen. While if we talk about site-specific recombination, it can target short DNA sites. There are many accessory proteins which are involved in case of uh, site-specific recombination. And both of these processes can be used for targeted gene alteration. Right? This is the essential thing. Now, site-specific recombination, it involves reciprocal exchange of DNA and it requires three main things. One is two DNA partners are required. One which would provide the new DNA, another one which will integrate this DNA, right? So there are two DNA partners which are important. A specialized recombinase proteins which will facilitate this process. It will recognize, it will cause DNA breaks, and then it will rejoin the DNA. So all this process are carried out by recombinases. And then there should be a mechanism inside the cell which would involve DNA breaks to happen and reuniting the DNA breaks using ligases, right? So these are the main three processes. If we look at some examples here, like we have already talked about lambda phages, which infect E. coli, and they causes integration of their genome inside the bacteria. That is one example of recombination. Another example could be transposons. Transposons like TN3 related transposons. These transposons are mobile genetic elements which can, travel between various genome sites, genomic locations. So essentially, let's say if here in this part of the genome there is some transposons, it can shift towards the other part at some other time. So this happens using recombination process. And then there is DNA inversion. It has been reported in case of Salmonella species and Salmonella species of bacteria, which causes various flagellar variations. So if you remember, in a bacteria, there is flagella. These flagellal variations are caused due to DNA inversion process. That also happens due to recombination. Another very important process, which you probably might have heard, is the VDJ recombination. If you have studied about immunology, it happens in, antibody generation. So VDJ recombination is one of the major reasons why immune system is programmed to recognize any antigen that comes to your body. So this happens due to, again, a recombination process, very important process, as a matter of fact. So is everything clear till now? Can I have a raise of hands, or maybe somebody can tell me? Yeah, cool. We'll move on. So site-specific recombination, it happens, I'm sorry. It happens due to recombinases. We already know. These are the enzyme. Now, these recombinases could be of two types. One is the tyrosine type. Another one is the serine type. Now, tyrosine type of recombination or serine type of recombination, this classification is based on the residue which catalyzes recombination, right? Uh, Now, this tyrosine type of recombination can be further classified based on the directionality. So it could be either unidirectional or it could be bidirectional. So unidirectional is, for example, phage integrase is an example of tyrosine type unidirectional recombinase. Bidirections are simple process. Since there are some simple tyrosine type recombinases. If we talk about serine type of recombinases, it could be further divided based on the protein size. So it could be either small or large. Very simple. So small would be resolvases, invertases. These are some of the recombinases which have both uni and bidirectional function. If we talk about phage integrases, which could be of serine type as well, they are usually large proteins. Okay. So tyrosine recombinases, they are found abundantly in prokaryotes, archaebacteria, eukaryotes, and even certain family of retrotransposons. Very conserved enzyme across the entire evolution. So the catalytic domain with recognizable sequence, motifs, and fold of the entire domain is conserved essentially. So what does this mean? That in this enzyme, the catalytic domain, essentially not only the domain, like not only tyrosine, which is the essential catalytic factor, which is the catalytic residue, not only tyrosine, but some other residues are also conserved across the entire evolutionary chain, right from prokaryotes to eukaryotes. It is conserved. So and it is also shared with two other class of enzymes. So two other class of enzymes also have a similar catalytic domain, similar to tyrosine type recombinases. One is type 1b topoisomerase, and then there is telomere resolvase, some group of enzymes, and protelomerase. So as we already know that topoisomerase are essentially important for relaxing DNA to unwind the supercoiling function of DNA. So we can say that the tyrosine type recombinases, these class of enzymes are also important there as well, right? In case of DNA unwinding, also telomeres are essentially ends of the DNA chromosome. So if you see of a DNA chromosome, this is how it looks like. The ends are called telomeres. So telomeres are usually resolved or their telomerase enzymes are there to protect these telomeres. And the presence of telomeres are very important because at every replication cycle, if this is, let's say, the DNA, at every replication cycle the DNA gets little bit shorter because a primer is added, and the primer is then removed due to exonucleases. So from 5' end, there is a little bit resection which happens after each replication cycle. Telomeres prevent DNA damage. So telomeres are basically excess of DNA. These are some repeats which are present at the end of the DNA. So they are present so that after each replication cycle, when DNA shortened, these telomeres would all get shortened, and it will prevent the essential genes which are present in the middle to get damaged. Right? This is how it works. And basically telomere shortening is also implicated in aging. So essentially in a eukaryotic cell, you already know that a cell can divide only a finite number of times. So in many theories, it is said until and unless your telomeres are active, they are present in the cell, in the DNA, and the telomeres can be shortened, at till that time there could be the process of DNA replication. Right after telomeres are completely shortened, there is no telomere present in the DNA. After that, the cell goes for apoptosis. And telomerases, which essentially causes telomeres to be maintained. These enzymes, telomerases, they are present in cancer cells, also in stem cells, which can replicate infinite number of times in theory. Well, so here you can see serine and tyrosine kind of recombinases, which are there. So in serine recombinase, it forms dimer, and you can see, the two DNA strands, they are joined in case of serine recombinases. In case of tyrosine recombinase, we see a holiday junction formation. So it essentially happens in a dimer only, but quadratic, I mean, two dimers are important for this process to happen. So there is a holiday intermediate, and it also forms a recombinant DNA. Right. Um, Some examples of tyrosine type simple recombinases are Cre recombinases and FLP recombinases. These are routinely used in experiments, in biological experiments. So if you have already heard about the Cre-LoxP system, mostly you have heard about it. It is used for creating conditional knockouts. So what happens is, let's say, if you're doing some experiment in mice models, and let's say if you want to knock out some gene and you do not want to knock out that gene right at the beginning, you want to knock it out at a later stage on a particular condition. So for example, let's say some particular gene X is responsible uh, for a particular disease, but if you knock out this gene X at right from the beginning of, let's say in case of mice, right at the beginning, it may cause neonatal fatality. That means it is very important in development of that mice from infant to adult. So if you just knock it out right from the beginning, it might happen that the mice dies. But you want to see how this gene X is important after the mice has developed as an adult. So there what we do, we introduce this gene X in a LOXP site, and there is the Cre recombinase, which is essentially the condition which we are giving. So Cre recombinase could be secreted at the later stage, and there could be several other ways of controlling it. There could be pH, there could be heat, there could be other things also which could regulate this Cre recombinase. As soon as Cre recombinase is produced, it will cause these LOXP sites to remove this gene, basically excision of this gene, and then this gene is removed. So you can find out if a particular gene can have some function in the later stage of the life of a mice. So for example, these are just examples. If we talk about applications, as I already told you about knock-ins and knockouts, sorry, knock-ins and knockouts. So knock-in is when a gene is inserted, right? That is called a knock-in. For example, let's say a bacteria which is ampicillin sensitive, which will die due to ampicillin. So you want to introduce ampicillin resistant gene into it. So that would be a knock-in, right? And once it is successful, this bacteria will survive in an amp plate, right? That is an example of knock-in. An example of knockout would be very similar, where a gene is removed, right? So let's say an important recombinase enzyme, a tyrosine recombinase enzyme. You want to remove this enzyme and see if tyrosine recombinases are absent in the system. What happens? So you could knock out that protein, right? These are some of the examples. Similarly, it is applied in the vast field of genome engineering, where you want to precisely manipulate genes in an organism. So this is all about the summary. You can read the slides for more details. Now, um, let's come to the question. What is termed as the guardian of genome integrity? Is it 1, cell division, 2, mutation, 3, DNA damage, or 4, homologous recombination? You can use the Q&A section to answer this question. Or you can even, just speak if you want to. It's an interactive session. I would like your answers. Nobody is responding.
Homologous recombination.
Yes, you're right. Homologous recombination is the answer. DNA ends resulting from a double-stranded break is processed by a multifunctional enzyme complex called the DREC-BCD, DNA polymerase, RNA polymerase, or gyrase.
Yeah.
Yes,
REC-BCD
REC-BCD.
complex.
Yes, right. You're right. REC-BCD complex is the answer. DNA polymerases, they cause polymerization of DNA. We know that. Similarly, RNA polymerase causes uh, polymerization of RNA. Gyrases are again proteins which are in the class of topoisomerase. They are also essential for coiling and uncoiling of DNA. Well, Chi site is the replication hotspot, DNA damage hotspot, recombination hotspot, or transcription origin.
Recombination.
Right, very, very, very right. They are recombination hotspot. Whenever a chi site is encountered, there these recombination events are more frequent. In classical non-homologous end joining or canonical non-homologous end joining pathway, which proteins act as the initial DNA end binding factor? Is it Q heterodimers, REC-BCD, XRCC4, or DNA PKCs? Anyone would like to answer? Yes?
Heterodymer. Yeah, sure.
Yes, right. You're right. You're right. Site-specific recombination between sites located on the same chromosome in a head-to-tail orientation results into excision, inversion, integration, or recombination. Any answers? So if there is recombination event, so let's say there is the orientation changes from head to tail. What would you call this event?
Inversion.
Right. A tyrosine type recombinase catalyzes 180-degree rotation and rejoining of cleave substrate DNA, formation of a holiday junction intermediate, single strand break repair or translation.
Formation of holiday junction intermediate.
Yes, yes. If we look at the slides before, I should have prepared. So they form a holiday junction intermediate. Integration of bacteriophage lambda into E. coli chromosome is an example of homologous recombination, non-homologous recombination, site-specific recombination, or DNA repair.
Site-specific recombination.
You're right. I don't know if I have it. Yeah. So if you see this example, what happens if there is a lambda phage and there is E. coli DNA? So lambda phage has P, then there is an O site. O is basically similar to a chi site. And then there is a P dash site, right? So P, because it's a phage, right? That's why it's P. Similarly, in case of bacteria, you have the B site, then again there is a O site, and there is a B dash site, which is B because it's from bacteria. Now, what happens is these two, they integrate something like this. So it will look like this. Please forgive my drawing if it's not good. Right. So here is the chi site. Now here we have the P dash and B dash. Okay. From here we will get something like this. So this the DNA of the phage, it gets integrated into the DNA of the bacteria. Can you see the drawing? I don't know.
No, sir. We are still in
No,
7th.
sir, not visible.
I'm really sorry. I don't know why this is. Okay. Trying to change the slides is not working. Let me just start presenting again. I think there is some network issue from my end. Can you still see it?
No, sir.
Yes? No, okay. Okay, I'll try rejoining. Just give me a moment. Okay, I'm not sure if you can see. Can you see it? No. Don't think so. I probably my iPad is not getting connected with the IIT Delhi's network. I'm trying to resolve this issue. Yeah, can you see it now?
Yes, sir.
So what I was saying is, uh, so yeah, so Prof, because it's a fudge, therefore it is P size, and we have a chi site in the middle, and then a P dash site, right? Similarly, for bacteria, we have a B site and a chi site and a B dash site. They form a complex like this. To integrate, and finally, the lambda profage element is integrated into the bacterial chromosome. So this is how it is a site-specific recombination. Hence, this process is also used for various gene editing as a gene editing tool. Fine. Question number 8. 180-degree rotation rejoining of cleaved substrate DNA is catalyzed by serine type recombinases, tyrosine type recombinases, both serine type and tyrosine type recombinases, or FLP recombinase.
Serine.
Yes, you are right. Serine type recombinases, they cause there is a phosphoserine linkage, so it causes cleavage of the the DNA strand, following which there is exchange by 180-degree rotation. So, and then there is ligation, where we see that the final DNA is now having like it is recombinated. Which of the following is true for the primary structure of REC-BCD enzyme? The N-terminal domain contains nuclease motif. The C-terminal domain contains nuclease motif. The C-terminal domain contains both nuclease and helicase motif, or the C-terminal domain contains helicase motif. Anyone would like to answer?
Option C.
Options B, actually. The C-terminal domain or the REC-C, it essentially contains only nuclease activity. Well, which of the following strategy a double-stranded break in which of the following strategies a double-stranded break present in one chromatid is repaired using its intact sister chromatid as a template? Homologous recombination, non-homologous end joining, alternative form of non-homologous end joining, or both NHEJ and A NHEJ? This is a very simple question. Most of you should be able to answer.
Option A, homologous recombination.
Right, right. You are absolutely right. Homologous recombination. In this case, we need an intact sister chromatid as a template. The Rec-C protein has 1122 amino acid residues and contains nuclease, chi recognition site, SF1 helicase motifs, or all of the above.
Chi recognition site.
You're right. So Rec-C recognizes. We've already discussed about it. Which of the following is not a part of Rec-BCD enzyme complex? Very simple. Rec-A is not a part.
Prot-
Yeah, correct. Which of the following enzyme is involved in prokaryotic polymerase non-homologous end joining component? Q proteins, the PE domain of Lig-D, the POL polymerase domain of Lig-D, or the LIG domain of Lig-D?
Option A.
In case of prokaryotes, I'm sorry, the POL domain is involved. Who proposed the eponymous Holiday junction model for DNA recombination intermediate? Was it Dr. Robin Holiday? Was it Dr. Rue Holiday? Was it Dr. Oliver Smithies or Dr. Oliver Holiday? On whose name was Holiday junction introduced? Anyone?
Dr. Robin Holiday.
You're right. Which family of enzymes do Cre and FLP recombinases they belong to? Is it tyrosine, recombinases, tyrosine integrases, serine resolvases or invertases, or serine integrases?
Tyrosine recombinases.
That's absolutely right. Lambda phase DNA contain an attachment sequence called dash, and bacterial DNA containing attachment sequence called dash. So is it ATTP and ATTB respectively, or ATTB and ATTP respectively, Rec B, Rec A, or Rec B, C, D, and Rec C?
Option A.
Option A.
Option A is the right answer. Which of the following is true regarding biological roles of for resolvases? The delta and TN3 resolvases are encoded by replicative transposons. Resolvases like beta and sine are encoded by Fat genome. Delta and TN3 resolvases are encoded by plasmids, or resolvases like beta and sin are encoded by replicative transposons. If you have read about resolvases, the first option is the right answer. FLP site is site-specific recombinase encoded by dash circle of the yeast Saccharomyces cerevisiae. Anybody would like to answer?
Option B.
Option B is the right answer. So that's all that we have for today. If you have any doubts, you can ask me right now.
Previous question, resolvases.
Yes.
Down.
Yes. What is the doubt?
A brief intro or hint of resolvases. What is its function like that?
Okay. So resolvases, in case of resolvases, there are three binding sites which are present. So, well, I'll also have to look at it once. So there is a delta site. So if you see the lectures, you will get it. Just give me a moment. So it is already given in one of the lecture slides that the delta and the TN3 resolvases, they are important for replicative transposons. So if you also remember that TN3 is important, they are basically transposons. These are elements, so essentially which can shift to multiple areas of the genome. So these resolvases, delta and TN3, they are important for this functioning of these transposons. I don't know if I made it any more clear, but it's part of the lecture.
Okay.
Any more questions? Okay, if that is all, we would like to end this session right here. Thank you all for joining. We'll be meeting next week. Let's stop the recording.